Good morning. Welcome back, everybody, to daily lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I am Dr. Bobby. We're on the subject of molecular biotechnology at the third section, and we're dealing with the following topics, namely genetic engineering of plants methodology, engineering plants to overcome biotic and abiotic stress, engineering plant quality and proteins, transgenic animals. And on to the matter proper. We just given an intro to the third section. Right? In the past, high-yielding strains of many different crop plants and farm animals have been successfully developed by selective crossbreeding. This time-consuming process has been superseded in part by the development of methods for the genetic engineering of higher organisms. Now, genes that contribute to specific traits can be introduced into plants and animals and then passed on from one generation to the next. How these transgenic plants and animals are formed and what sorts of traits are being manipulated are examined in part 3. A number of transgenic plants have been engineered to be able to overcome a variety of biotic and abiotic stress or stresses, including insect predation, viral infection, herbicides, pathogenic fungi and bacteria, oxidative stress, salt stress, and drought. Transgenic plants that have a significantly improved nutritional content produce fruit or vegetables with enhanced taste or appearance, or produce flowers with altered pigmentation have been created. In addition, some transgenic plants have been used to facilitate environmental cleanup, phytoremediation, while others have been used as living bioreactors in the production of a range of foreign proteins, including many therapeutic agents. Transgenesis of animals has included studies of cattle, sheep, goats, birds, and fish, all based on the principles that have been established for creating transgenic mice. Some of these transgenic animals have been developed to produce therapeutic proteins in their milk or eggs. We're now on to the topic of genetic engineering of plants, methodology in specific. Topics that are covered under these are plant transformation with the typlasmid of atumifacients, typlasmid-derived vector systems, physical methods of transferring genes to plants, microprojectile bombardment, chloroplast engineering, use of reporter genes and transformed plant cells, manipulation of gene expression in plants, isolation and use of different promoters, gene targeting, targeted alterations in plant RNA, facilitating protein purification, production of marker-free transgenic plants, removing marker genes from nuclear DNA, removing marker genes from chloroplast DNA, summary and review questions. Right, on to the first chapter on the third section. I repeat, genetic engineering of plants, methodology. Now, considerable effort has gone into developing varieties of plants that produce increased yields and have enhanced nutritional value. Although much of this endeavor has been directed toward the three major gains, corn, maize, wheat, and rice, successful breeding programs for other food plants and horticulture species have also been established. Recombinant DNA technology, which has been used extensively in microbial systems, is also an important tool for the direct genetic manipulation of plants. There are a number of effective DNA delivery systems and expression vectors that work with a range of plant cells. Furthermore, most plant cells are totipotent, meaning that an entire plant can be regenerated from a single plant cell, so fertile plants that carry an introduced gene or genes in all cells, that is, transgenic plants, can be produced from genetically engineered cells. If the transgenic plant flowers and produces viable seed, the desired trait is passed on to successive generations. There are three major reasons for developing transgenic plants. First, the addition of a gene or genes often improves the agricultural, horticulture, or ornamental value of a crop plant. Second, transgenic plants can act as living bioreactors with an inexpensive production of economically important proteins or metabolites. Third, plant genetic transformation transgenesis provides a powerful means for studying the gene action of genes during developmental and other biological processes. Some of the genetically determined traits that can be introduced into plants by a single gene or possibly a small cluster of genes are insecticidal activity, protecting against viral infection, resistance to herbicides, protection against pathogenic fungi and bacteria, delay of senescence, tolerance of environmental stress, altered flower pigmentation, improved nutritional quality of seed proteins, increased post-harvest shelf life, and self-incompatibility. In addition, transgenic plants can be made to produce a variety of useful compounds, including therapeutic agents, polymers, and diagnostic tools such as antibody fragments. Alternatively, they can be engineered to synthesize viral antigenic determinants and after ingestion can be used as edible vaccines. To date, over 150 different plant species have been genetically transformed, including many crop and forest species. We're on to a milestone note now. This is the debate over genetically modified foods effectively over. The details of that. Rice is the most important crop in the world. In 2004, China, the world's largest producer of rice, announced a ramping up of efforts to commercialize genetically modified rice. India, the second largest producer of rice, was quick to follow suit, with two largest rice producers in the world. With 40% of the world's population switching to genetically modified rice, the rest of the world will eventually have little choice but to accept genetically modified foods. From 1996 to 2007, the global area devoted to transgenic crop has increased from 1.7 million to 114 million hectares. In 2007, the major producers of transgenic plants were the United States, 57.7 million hectares planted, Argentina, Brazil, Canada, India, China, Paraguay, South Africa. Major transgenic crops worldwide included, in order of a number of hectares planted, soybean, corn, cotton, canola, rice, squash, papaya, alfalfa, wheat, and eggplant. Major traits that have been introduced into plants include herbicide tolerance and insect resistance. In 2002, the Food and Agriculture Organization, United Nations, endorsed the development and use of genetic modified foods. According to researcher Florence Wambugu of Nairobi, Kenya, the African continent urgently needs agriculture biotechnology, including transgenic crops, in order to improve food production. Famine provides critics with an opportunity to promote an antibiotic message that only results in millions of people who urgently need food starving to death. Wambugu urged the public to recognize the difference in needs between Europe and Africa. Europe, with a population that is under control, has surplus foods and does not experience hunger, whereas Africa encounters experience mass starvation and death by mid-2008. And South Africa was the only country in Africa to have approved the commercial use of genetically modified crops. And over that, into the main subject. In over 50 countries worldwide, this was seen. Plant biotechnology is having the enormous impact on plant breeding organisms because it significantly decreases 10 to 15 years that it takes to develop a new variety using traditional plant breeding techniques. By mid-2008, researchers had reported the complete DNA sequence of hundreds of microorganisms and dozens of animals, but only 3 plants: Arabidopsis thaliana, rice, and poplar. At that time, the genome sequencing of several other plants, including corn, soybean, canola, tomato, cotton, potato, cassava, sorghum, grape, and peach, had been initiated, while the study of plant genes and genomes clearly lags behind studies of animals. It is gaining momentum. So that within the next 5 to 10 years, a wealth of information with an enormous impact on plant biotechnology is expected to become available. Despite all of the programs, right? So, despite all the progress that has been made in the development of transgenic plants for a wide variety of purposes, a vocal minority of individuals in the United Kingdom, in North America, and a large number in Europe still oppose the use of this technology. Nevertheless, with each succeeding year, since the mid-1990s, the use of transgenic crops has continued to increase both in absolute terms and in the number of countries using this technology. It is expected that in the not-too-distant future, the majority of agriculture crops worldwide will be transgenic. Plant transformation with the type plasmid of A tumefaciens, the gram-negative Solbacterium. Agrobacterium tumefaciens is a phytopathogen that as a normal part of its life cycle genetically transforms plant cells. This genetic transformation leads to the formation of crown gall tumors, which interfere with the normal growth of an infected plant. The agronomically important diseases affects dicotyledonous plants. Dicots includes grapes, stone fruit trees, example peaches and roses. This genetic transformation leads to the formation of crown gall tumors, which interferes with the normal growth of an infected plant. This agronomically important disease affects only dicotyledonous plants, dicots, including grapes, stone fruit trees, some peaches and roses. Crown gall formation is the consequence of the transfer, integration, and expression of genes of a specific segment of bacterial plasmid DNA. Called the tDNA, transferred DNA into the plant cell genome. The tDNA is actually part of the tumor-inducing TI plasmid that is carried by most strains of A tumefaciens. Depending on the TI plasmid, the length of the tDNA region can vary from approximately 10 to 30 kilobase pairs. Strains of A tumefaciens that do not possess a TI plasmid cannot induce crown gall tumors. The crown gall formation is the consequence of the transfer, integration, and expression of genes of a specific segment of bacterial plasmid DNA, called the tDNA. And depending on the TA plasmid, the length of the tDNA region can vary from approximately 10 to 30 kilobase pairs kb. Strains of tumefaciens that do not possess a TA plasmid cannot induce crown gall tumors. The initial step of the infection process is the attachment of tumefaciens to a plant cell at the site of an open wound, often at the base of the stem, that is the crown of the plant. After the initial attachment step, a tumefaciens produces a network of cellulose fibrils that bind the bacterium tightly to the plant cell surface. Originally, it was thought that A tumefaciens infected wounded plants because the physical barrier of the cell wall had been breached by injury, thereby facilitating entry of the bacterium. However, it is now recognized that these bacteria respond to certain plant phenolic compounds such as acetylsyringone and hydroxyacetylsyringone, which are excreted by susceptible wounded plants. These wounded response compounds resemble some of the products of phenylpropanoid metabolism, which is the major plant pathway for the synthesis of plant secondary metabolites such as lignans and flavonoids. These small molecules, that is acetylsyringone and hydroxysyringone, act to induce the virulence VIR genes that are carried on the TI plasmid. The VIR genes is located on a 35 kilobase per region of the TI plasmid that lies outside of the tDNA region. There are 25 VIR genes arranged in 7 operonds in the plasmid. The products of the VIR genes are essential for the transfer and integration of the tDNA region into the genome of a plant cell. After a TI plasmid carrying cell of A tumor patient attaches to a host plant cell and the PIR genes are induced, the tDNA is transferred by a process that is similar to plasmid transfer from donor to recipient cells during bacterial conjugation. In this model, the tDNA is transferred as a linear single-stranded molecule. The TI plasmid enters the plant cell and eventually becomes integrated into the plant chromosomal DNA. The formation of the single-stranded form of the tDNA is initiated by strand-specific cutting by an enzyme encoded by one of the VIR genes at both borders of the intact tDNA region. The 5' end, the single-stranded tDNA carries the red border sequence, and the left border sequence is at the 3' end. The integration of the tDNA into the plant genome depends on specific sequences that are located at the right border of the tDNA. This border contains a repeating unit that consists of 25 base pairs. Although the left border contains a similar 25 base pair repeat, deletion studies have shown that this region is not involved in the integration process. During the insertion of tDNA into plant chromosomal DNA, short deletions of the plant DNA are often produced at the junction between the tDNA and the plant chromosomal DNA. In addition, while the insertion of the tDNA into the plant DNA occurs at random sites, the tDNA borders exhibit some homology with the plant DNA at the site of insertion. Most of the genes that are located within the tDNA region are activated only after tDNA is inserted into the plant genome. This reflects the fact that these are essentially plant genes, which cannot be expressed in the bacteria because of differences in transcriptional and translational regulatory genes or sequences between the two types of organisms. The products of these genes are responsible for crown gall formation. The tDNA region includes some genes RAA, M, and RAAH. These pair of genes encoding enzymes that synthesize the plant hormone auxin indole acetic acid. Specifically, IAM codes for the enzyme tryptophan 2 monoxygenase, which converts tryptophan to indole 3-acetamide, and IAH encodes indole 3-acetamide hydrolase, which converts indole 3-acetamide to indole acetic acid. In addition, the tDNA region carries the TMR gene, also known as IPT, which encodes isopentenyl transferase. This enzyme adds an isopentenyl side chain to a 5' AMP to form isopentenyl adenosine 5' phosphate, the first committed step in the synthesis of cytokine isopentenyl adenine. Hydroxylation of these two molecules by plant enzymes generates the cytokines called transziatin, transribosylase, respectively. Both auxins and cytokines regulate plant cell growth and development. In excess, they can cause the plant to develop tumorous growth such as crown galls. In addition to auxin and cytokine biosynthesis genes, the tDNA region from each specific TI plasmid carries a gene for the synthesis of a molecule called an opine. Opines are unique and unusual condensation products of either an amino acid and a ketose acid, or an amino acid and a sugar. For example, the condensation product of arginine and pyruvic acid is called octopine. Arginine with alpha-ketoglutaraldehyde is nonylpalene, and agropine is a bicyclic sugar derivative of glutamic acid. The opines are synthesized within the cell gall, the crown gall, and then secreted. They can be used as a carbon source and sometimes also as a nitrogen source by any A tumor patient cell that carries a diplasmid-borne gene for the catabolism of the particular gene. The opine catabolism gene S is on the Thai plasmid and is not part of the tDNA region. All other soil microorganisms that have been tested are incapable of utilizing opines as a carbon source. There's a unique set of mechanisms has evolved whereby each strain of its tumefacients genetically engineers plant cells to be biological factories for the reduction of a carbon compound that it not alone is able to use. Thai plasmid-derived vector systems.